Okay. Okay. So now already 8 o'clock. Okay. I will start the live now. So for today, is some one class. Sorry. For today is will continue to the semester one class. So I hope everyone can hear my voice. Okay. So I think everyone can hear. My mic is open. Okay. So for today, we will end our teach all the sem one class, which is what nucleic acid, and also the last topic, analytical method. Okay, so for nucleic acid, so actually I think most of you are familiar with it, right? So acid is our DNA, right? So actually, if you see drama, actually got tell about more about DNA, right? So actually, nucleic acid who found the nucleic acid. So actually, is this scientist? This scientist found the found it is the scientist, the first person in the world who who founded the nucleic acid structure. So the name is Friedrich Michel in 1869. So this is just the history lah. Actually, you don't need to know. Okay. Okay. So what is nucleic acid? Okay, so nucleic acid we can define as it is the polymer structure that consists of nucleotide residue. So actually nucleic acid is the common name lah, common name for the polymer like carbohydrate, like protein like that. And it is the common polymer, and it is made up of nucleotide. So the monomer of the nucleic acid is nucleotide. Okay, so normally the nucleic acid is located in the nucleus of the cell. Okay, so it is at the nucleus of the cell, but this is for eukaryote lah. But for the prokaryote, actually prokaryote does not have nucleus, so actually it is will lie at the cytoplasm lah. Because prokaryote does not have nucleus, right? So it just lie on the cytoplasm, so it is not what we call as net DNA lah, because it does not have the nuclear envelope to bound the nucleic acid one. Okay, so normally the elemental composition is carbon, hydrogen, oxygen, nitrogen, and phosphorus. So normally the function is store genetic storage, store the genetic information. It also can transfer the genetic information and also energy transfer. So this is just the general function lah. Okay, so actually you guys know that we have two main types of nucleic acid only, which is DNA or RNA. Okay, later we will talk detail about it. So now we will proceed to the monomer of nucleic acid that is called as nucleotide. Okay, so what is nucleotide? So actually, if you see this diagram, this is the basic structure of nucleotide. Yeah? Okay, so this is the fundamental building block of nucleic acid. It's just like the carbohydrate, lipid, and protein. It also have monomer, right? So like carbohydrate, the monomer is monosaccharide. Like for the lipid, the monomer has two, right, glycerol and fatty acid. And then for protein, the monomer is amino acid. Same as the nucleic acid, it also have monomer. The monomer is nucleotide. So this is the structure of nucleotide. You can see that it contains three main subunits. So the first subunit is pentose sugar. So it is the 5 carbon sugar. It only have 5 carbon only. So you can see 1, 2, 3, 4, and 5 at here. So it is 5 carbon sugar, and then another one is nitrogenous base. So this one nitrogenous base is based on the type of the nucleotide. So as you guys know, we have five types of nitrogenous base, right? Adenine, thymine, cytosine, guanine, and uracil. So these are the nitrogenous base. Based on this is the differentiate the type of the nucleotide. So we have 5 types of nucleotide, right? And then this one is phosphate group. So this one is almost the same. All of the nucleotide have this phosphate group, pentose sugar, and also nitrogenous base. Okay, so when we talk about the pentose sugar, so this is the structure of pentose sugars. Okay, so it is the five carbon structure, and if you alert for it, the carbon number two, it can be either hydroxyl group or only hydrogen atom attached to it. So this one, that is, we differentiate between the DNA and also RNA. So why we call DNA as DNA? Because of deoxyribose. Deoxy, meaning that the C number two at here does not have the oxygen atom. So that is why we call as deoxy. Dia sudah terminate. The oxygen is terminated at there. No oxygen at the carbon number two. But for the ribose, it contains hydroxyl group. So it contains oxygen atom at carbon number two. So that is why we call RNA. So ribose. So this is actually as the original structure of ribose. Original structure of ribose should be have the oxygen atom hydroxyl group one. But for the DNA, it does not have the hydroxyl group, so we did not naming as ribose. So we naming as deoxyribose. That is why DNA, the DNA called DNA. The D represent the deoxyribose. The R represent ribose only. So this is one of the main differences. Okay. Next is about the phosphate group. Okay, so actually the phosphate group is the PO42 negative inorganic compound. So it is the polar molecule and it is highly ionized oxygen atom. So you can see that it has contain 4 oxygen atoms. So it is very highly ionized one. And also impact negative charge to the phosphate group. So this is why we say that the nucleic acid is negatively charged because of this phosphate group. If you got study the gel electrophoresis, later I will explain. You already learned, right? Actually, you already learned that DNA is negatively charged. So why it contribute negatively charged? Because of this phosphate group. Because this phosphate group got contain 2 oxygen atom, which is contain lone pair electron one. So that is why that the DNA and RNA molecule is contribute to negatively charged because of this, because of phosphate group. Okay, next is about nitrogenous base. So just now I just mentioned that we have 5 types of nitrogenous base, right? So actually we can divide it into 2 class of nitrogenous base. Either it is pyrimidine or purine. So what are the difference is the structure. So actually if you see that pyrimidine only contain the hexane, what we call cyclohexane structure only. Cyclohexane. So what is cyclohexane? For chemistry senior, you should know. This is the structure we call as cyclohexane. So or in general name we call as 6 ring lah. 6 carbon ring structure. 6 carbon ring hexagon lah. Hexagon structure. And for the purine, it contains 2 ring structure. One is hexagon and another one is pentagon structure. So it contains two ring structure that bound together. So this one we call purine, this one we call pyrimidine. So normally purine only have 2, and either adenine and guanine is under purine. Thymine, cytosine, and uracil are under pyrimidine. So the another difference is pyrimidine conform smaller, right? And purine is more larger lah, because purine is more complicated, right? Compared to pyrimidine. And then the detailed structure is at here. So these are the differences between them. So normally the function is to donate electron and form new molecules. So this one is the key molecule that differentiate the nucleotide. So altogether we have 5 nucleotides. So normally, but DNA and RNA only have 4 nucleotide present. So you will see and also thymine is the key differences between DNA and RNA lah. So UACL will only present in RNA, and for the thymine only present in DNA only. So these are one of the key difference between DNA and RNA. Okay, so polymer. Okay, I don't want to mention already. What is a polymer? Actually, is made up of very monomer linked together, become large molecule lah. Okay, so polymer nucleic acid is polymer or nucleic acid is is the long chain of nucleotide lah. And then the nucleotide undergo condensation reaction, actually same as the previous biological molecule, not water, carbohydrate, lipid, and also protein. The same process only. And what are the two main class of nucleic acid? DNA and RNA. So the bond between the nucleotide is we call as phosphodiester bonds. So these are the specific name of the bond between the nucleotide. Okay, so phosphodiester bond are the covalent bond between the pentose sugar of one nucleotide and phosphate group of another nucleotide. So if you see here this structure, the phosphodiester bond is here, and also here, and here. So I highlight here. So this one actually is we call as phosphodiester bond lah. So it actually is bonded with one carbon at here, and also another one is phosphate group. They are bond together. It bond together. Bond together. Not here. Here. Sorry. Phosphodiester bond is here. Sorry. This one is this one is the basic structure of nucleotide. This one, yang ni, tengah punya, straight punya, yang ni. This one is a more phosphodiester bond. So if in exam, the person ask you to draw the dimer dimer of the nucleotide, so you have to draw like this. Don't need to draw all four lah. Actually, you just only draw the upper part 2 is enough already. Because the examiner normally want to test you either you know the location of the phosphodiester bond or not. So phosphodiester bond is normally bonded by 1 to C number 3, carbon number 3 one. It bonded with carbon number 3 and another oxygen atom of the phosphate group. Okay, so you should know this. Okay, so two main class of nucleic acid, DNA and RNA. So one of the differences between them is DNA contain 2 polynucleotide chains. The polymer of the nucleic acid we call as polynucleotide. So DNA contains 2 strands of polynucleotide, but RNA it only contains one polynucleotide chain only. This is one of the main difference. DNA contains 2 polynucleotide chains, but RNA only have 1 polynucleotide chain. Okay, so DNA normally is double helical structure. It will form a double helical structure one. So why we call double? Because of 2 polynucleotide chains, they bonded together between the nitrogenous base, and then to form double helical structure chain. And so it contains presence of hydrogen bond between the nitrogenous base. So between the nitrogenous base, it contains nitrogen bond. But compared to RNA can form helical structure also, but it only does not have the hydrogen bonding between another polynucleotide chain. It has hydrogen bonding also, but the hydrogen bonding is among the same nucleotide chain, but it does not have the nitrogen bonding between another polynucleotide. So actually, you will see the structure of RNA lah. Okay. So now DNA. So actually, when we talk about DNA, actually the DNA the structure is actually discovered by the Crick and Watson model. So these are the two scientists lah, Crick and Watson. Tapi who is Crick, who is Watson, I don't know. I already forget. So actually they need to know Crick and Watson. So these are the two scientists that discovered and then they found that DNA structure is look like that. Uh, okay, so actually DNA structure I already say it is the double polynucleotide helix structure. So it is a helix structure consists of two polynucleotide chains that strand coil together and complementary to each other. So what is we mean by complementary to each other? This is what we mean at here. So normally, the bonding between the nitrogenous base is based on between the nitrogenous base is already fixed one. It cannot be changed one. Dia bukan suka hati or adenine dia suka-suka nak bonded with guanine. Dia suka-suka nak bonded with cytosine. No. It is already fixed. So what is mean by complementary? Because it is already fixed. So adenine will be fixed to the thymine one. Conform. Either the adenine is fixed to the thymine or adenine can be fixed to the uracil. Either cDNA or RNA. If DNA is thymine, if RNA is uracil. So for others, cytosine only can be fixed with guanine only. Cytosine, this one is cytosine. Cytosine only can fix with guanine only. C and G. So A, B, C, G. I normally I memorize like that. A, B, C, G. So A is bonded with T, C is bonded with G. Okay, so and then why does the differences because of bonding? So adenine bonded with thymine contain 2 hydrogen bond, but cytosine bonded with guanine, it contains 3 hydrogen bond. So that is why only adenine can bonded with thymine or uracil, because both of them can only form 2 hydrogen bond. And cytosine and guanine can form 3 hydrogen bonds, so they bond together. So that is what we call as complementary. Okay, and another one structure you can see that the DNA structure is anti-parallel between the both chains. So if the structure, this one is 5 to 3, this one 5 to 3, and then for another strand will be anti-parallel, meaning that upper one will be 3 direction and lower one will be 5 direction. So what does this 5 and 3 mean? So this one is the carbon number one. So this one will be ended with carbon number 5. This one is ended with carbon number 3. So automatically this one is anti-parallel. So the upper one is ended with carbon number 3 and lower one is ended with carbon number 5. So if the exam want you to draw the structure like this, maybe it can come out want you to draw the structure like this. So you have to alert the hydrogen bonding and also the direction of the polynucleotide change, because both of them are anti-parallel. If you draw the structure which draw the structure 5 to 3 ball or 3 to 5 ball which is same direction, meaning you are wrong already, because DNA is anti-parallel. So you have to alert for it. You have to know that when you draw this 5 to 3, meaning that this one will be 3 to 5. And then the hydrogen bonding, also adenine and thymine only have 2 hydrogen bond. Cytosine and guanine have 3 hydrogen bond. Okay. So So that is what I just mentioned, anti-parallel. And then they are linked together by phosphodiester bond. And then they have the repeated sequence of the phosphate deposit ribose form the sugar phosphate backbone. Okay, and then 2 polynucleotide chains are held together by hydrogen bond between nitrogenous base. So for this, Okay, for this, actually, what is meant by sugar phosphate backbone when they are when the DNA structure, you see this, when the DNA structure is bonded together, bonded together, they will form the sugar phosphate backbone like that. So that is why it look like straight line. Two DNA structure is straight line. So this is what we call DNA ladder structure. But actually, original DNA will not have this structure. Original DNA in our cells is the structure like this, double headed structure, like this one. The original DNA structure is not like that. This one is actually we already break the double headed structure become two straight polynucleotide structure. And then when we more detail, this one is the and during the examination you should draw like this. So if the exam want you to draw, you should draw the structure like this. Not this and not this. You have to draw the structure like this. So this is what we call sugar phosphate backbone. Actually, if you compare to this structure, actually this whole structure is we call as sugar phosphate backbone. Why we call backbone? Dia macam backbone lah. Backbone, our backbone that provide the strength and support to the DNA one, to the nitrogenous base. So they are look like backbone. So what we call the sugar phosphate backbone. Sugar is present of pentose sugar, phosphate is a phosphate group, and then backbone because it look like the backbone that provide the support to the nitrogenous base. So finally it will look like that lah, double helix structure. So it is the original DNA structure that look like. Okay, now I already explained all about DNA. So I hope no question. Okay, so actually you have any question, you just type in the chat box. Okay, I will alert about it. Okay, so these are all about the DNA structure. So actually DNA not hard. Actually very easy to learn because it only have one type. But for RNA, the main difference I already mentioned is the nitrogenous base. So RNA, the nitrogenous base is thymine is replaced by uracil. So adenine, cytosine, and guanine is normal, are also present in the RNA, but the thymine is not present in RNA. It is replaced by uracil. And RNA only have single polynucleotide change only. Compared to DNA have double. RNA only single. Okay. Okay, so there are three main types of RNA you have to learn. So the first type is mRNA, messenger RNA. Second type is ribosomal RNA, rRNA. And another one is tRNA, transfer RNA. Okay, so both these three you have to know their structure and also their function. Okay, so messenger RNA. So messenger RNA is the mRNA. The short form is mRNA. So mRNA look like this. It looks like DNA, but actually not. It is the heading structure, but it only have single polynucleotide change only. Okay, so normally mRNA is only single RNA nucleotide polymer change and contain complementary codon formed by the transcription of a single cistron gene on a single DNA strain and carry genetic code transcript from DNA in the nucleus to the ribosome in the cytoplasm where protein is synthesized, form about 3 to 5% of the total RNA of the cell. So in general, mRNA is the normally it will present through the process called transcription. So in general, because you are only SEM 1, actually transcription and translation process you will learn more detail in semester 3. So for their senior at here, actually they will learn about that. Okay, so actually I just talk in general about the central dogma theory. Okay, in our body, our cells it can produce the protein molecule. So how our cells produce protein molecule, it will based on DNA undergo through the transcription process produce mRNA, and then mRNA will produce through the translation process to produce a protein molecule. So the process you don't need to know, you will learn in semester 3, you don't need to know. You just know that transcription is the process that come from DNA one. It produce mRNA come from DNA, using the information of DNA to produce the mRNA. So normally mRNA is shorter compared to DNA because not all the information from the DNA will be transcribed to the mRNA one. Only the certain specific information that bound with the protein will be transcribed. So that is why form about 3 to 5% only. So not all the DNA dia tak copy semua. You can say that it is copy, not copy semua DNA punya info kepada mRNA1. No, it only copy some important genetic code that is bound with the protein only. Okay, so contain complementary codon, so it has the complementary codon with DNA one. So if the DNA here is A, maybe E is A, and then the mRNA here will be T, will be U, not T, because RNA does not have thymine, will be U. And if the DNA here is C, and then this one will be G. So it is complementary to the DNA lah. Okay. So the main function of the mRNA is to carry the genetic code that transcribed from DNA, and then it will go to the ribosome. mRNA will go to the ribosome, will undergo translation process to produce protein molecule. So the mRNA main function is just carry the genetic code only, the genetic code that transcribed from DNA only, just like that. The main function of the mRNA. Okay, now about rRNA, ribosomal RNA. So actually, if you see this structure, why the structure like that? Okay. Okay, so ribosomal RNA. So if you see that ribosome, so it is the RNA that locate within the ribosome one. So this is the ribosome structure, right? A small unit and large unit. So rRNA is located within the ribosome structure. So ribosome it contains protein and RNA, right? So RNA is within the ribosome structure. So the main function is to aggregate with protein to form the large ribosomal subunit and form small ribosomal subunit. So in general, it is the component of the ribosome unit. So if you want to form the ribosome structure, RNA is one of the important component to form it. If no RNA, no ribosome. So it is the most abundant form of RNA in the living cell because all of the cells, either eukaryote or prokaryote, contains ribosome. Right? If you know the differences between prokaryote and eukaryote, all of the cells, either you are prokaryote or eukaryote, it contains ribosome. Even the virus also, some certain virus also contain ribosome structure. Some, not all, some. Okay, and then the sequence RNA have been used to characterize the evolutionary relationship between organisms. So this is the latest finding lah. They found that if they found the sequence of the RNA, actually we can learn the variation of the organism. But between the organism, we can based on that learn about the variation between them, and consists of the polynucleotide chain. So same, and it is synthesized by nucleus also in the nucleus. So after it is synthesized in the nucleus, then they combine with the protein molecule to form the ribosome structure. So the primary function is in protein synthesis. It also involves protein synthesis because ribosome actually is a unit that play important role in protein synthesis, right? Although I am not touching the chapter 2, but I know that your school teacher, I think your school teacher is already Now chapter 3, so you should know the function of ribosome. If you don't know, never mind, I will teach you tomorrow. Okay. Okay, so the last type of RNA is transfer RNA, tRNA. So if you look the structure of tRNA is very different from rRNA and mRNA one. Uh, you can see that it is look like the loop handle structure. This one structure we known as loop and handle structure. LOOP and handler. HENLE. HENLE. Okay, yeah. HENLE structure. So these are the spatial structure of tRNA. So if you see that tRNA, so it is the single strand polynucleotide folded into a three-dimensional structure called clover leaf shape, which is held by hydrogen bond. So although it is just the single polynucleotide chain, but it can form the 3D structure. So 3D structure, and then how they form, it is bonded between the hydrogen bond. So you will see here there are many hydrogen bonds here that held together. So this one just is a hydrogen bond that bonded together to form the 3D structure. And then if you see here, the 3' end always end in the base sequence of CCA and act as the amino binding site. So if you see here, the 3' end of the polynucleotide chains here, it will be ended with CCA lah. Normally CCA, and then it is bounded with the amino acid. So if you know that the main function of the tRNA is carry amino acid for protein synthesis during translation process. So what is translation? I already mentioned here. So translation is mRNA to protein. Is the process that we use mRNA for information of mRNA to translate the information translate the information of mRNA to produce protein. But not only one mRNA, not only mRNA, tRNA is the key molecule that carry amino acid. Dia bawa amino acid tu kepada ribosom and then hasilkan protein. So protein ni the amino acid, semua amino acid, we know that the monomer of protein is amino acid, right? So how amino acid is come from? All of the amino acid is come from the tRNA one. And tRNA, because tRNA need to ensure that all of the amino acid bounded together is true one. Not sesuka hati, tak sesuka hati amino acid nak bound together. No. tRNA is dia macam teacher lah, guide the mRNA so that the protein is bounded exactly based on the information for mRNA. So you can say that tRNA macam cikgu lah, dia macam guru, and then mRNA dia macam syllabus. Syllabus, based on the syllabus, okay, guru cikgu awak ajar, okay, ajar macam saya ajar kamu, ajar to make sure that your students are go to the right direction to help you ensure that your knowledge obtained is true, not false. So if the teacher go to the false direction, tRNA got something problem at there. So normally the protein structure will be teenager because they already have something wrong at there. So the tRNA is a key molecule that bonded, they can bind with the amino acid. So which amino acid it will bind? Because we know that there are 20 types of amino acid, right? In the world, got 20 types of amino acid. So which amino acid will bind to it? Methionine, lysine, lysine, or aspartic acid, or and others. So which amino acid bounded to this tRNA is based on the anticodon. This one, anticodon at here. This one, if you see here, this structure, these three base pair, the loop and this structure at the bottom. have 3 base pair we call as anti-codon structure. So these are the triplet structure, triplet base structure that is directly related to the specific amino acid carried by the tRNA molecule. So this is the key structure that determine what amino acid bind to the tRNA. So if okay, so these are the key molecules. So when we say there are 20 types of amino acid, meaning that tRNA should be have at least our body have at least 20 types of tRNA. 20 different types of tRNA because they need to bound with amino acid one. But not all the cells can produce all 20 types of amino acid. Like human, we know that we have essential and non-essential, right? Non-essential is that we can I think essential amino acid and non-essential amino acid, right? So normally our human only can produce only can own synthesis non-essential only. Essential we need to consume, right? Okay, if I'm not mistaken. Right. So maybe you can check for it. So normally in human, you only can synthesis 11 amino acid. Another 9 amino acid is need to be carried by but however, our DNA should be have 20 types, however, because they have to bound with different 20 types of amino acid to form the protein molecule. Okay, so actually this is all about DNA. So the main function of DNA is just the carry the amino acid during the translation process to produce protein, like that. So actually this one normally if you first time here you can be very complicated, many question mark, because this one you will learn more detail in semester 3. So in semester 3 you should know lah the process between the transcription and translation. Once you know the transcription and translation process, then you will know the function about the mRNA and tRNA. But for now, because you are not semester 3, so I am not allowed to teach you, because if I teach you, then later you are kepala besar lagi. You will be very headache. You will have headache. So I just give you the simple knowledge only. So transcription is DNA to mRNA, translation is mRNA to protein, and then with the help of a tRNA, just like that. You just want to know like that. Okay. Okay, so the differences between DNA and RNA, I think I don't need to talk much because I already mentioned many times. So about the location, this one I not mentioned yet. Location, normally DNA is found on the nucleus of the cell and also in the mitochondria and chloroplast. Chloroplast and mitochondria got DNA, right? Got DNA one. But for RNA, normally it only found in cytoplasm. It can be found in cytoplasm, can be found in nucleus, and it also can be found in ribosome. So got three locations lah: cytoplasm, nucleus, and ribosomes. Okay. But for DNA only can found either it is in nucleus or in the mitochondria or chloroplast. Okay, so for the definition, I think I don't need to explain more. Just the main difference is the thymine and uracil. Thymine is replaced by uracil for RNA, and for DNA it is the long polymer. It is more longer, but RNA is more shorter. And then the sugar portion, the carbon number 2, right? Number 2, DNA does not contain oxygen atom, so it is we call as deoxyribose. Okay, next one is about function. So normally function of DNA function is the transmission of genetic information. It carries genetic information. DNA is the molecule that carry genetic information. But RNA, sorry, it does not contain any genetic information. It just functions as the transmission of genetic code that is necessary for protein synthesis, for nucleus to the ribosome. So you have to just to know that RNA normally is function for protein synthesis. It just copy information for DNA and then transcribe or transmit the information from the DNA to produce protein, just like that. And predominant structure, so the structure of DNA is normally double-stranded, RNA is just single-stranded. And propagation, okay, so DNA can be replicated, can undergo replication at its own. And actually propagation DNA RNA can be replicated one. Typo it should be RNA. Okay, you ultraviolet damage. So DNA normally is more sorry, more easily to be that be destroyed by ultraviolet UV light, but compared to RNA is more resistant to UV light. Yeah. Okay, so I think that's all for for nucleic acid. Okay, so any question about nucleic acid? I think no, right? Because it's very easy, right? So no question. I see this no question at all. Okay, so I assume everyone know already. What about the nucleic acid? If you have any question, just type. If I see, I will answer it. Okay, so next I will proceed to the analytical method. Analytical method. Okay, so I think this topic normally you guys know. Take more effort, right? Because normally if you see the Campbell book, I think Campbell does not mention paper chromatography, right? And then gel electrophoresis, even in Campbell, you see that the information is very wide, very spread away. You even don't know which parts mention gel electrophoresis. So actually, this one analytical method actually is more about the separation technique lah. Analytical method, what is mean by analytical? We mean that we need to analyze something. So in NPM, they are letak syllabus at chapter 1, meaning that we need to analyze biological molecule. So normally we analyze mainly two main structure, which is DNA and nucleic acid. So because these two biological molecules okay, okay. Okay, I just now I thought okay wait. Oh, okay. Normally in biology, in nowadays, scientists is more interesting the information in DNA, nucleic acid, and also protein. Why? Because nucleic acid and protein are the more biological molecule are very complicated. Because every organism you imagine, but I just say nucleic acid each made different types of species, different types of organism will have different types of DNA sequence and RNA sequence, right? Like human DNA sequence is not same like dog. Even dog have many species, right? The different species between dog also have the differences with the nucleic acid structure, DNA structure, and also RNA structure. So when the structure is different, automatically the protein structure also different because protein structure and genetic information is very they are related together because protein is synthesis by DNA, right? The protein structure is it is synthesized based on the information from DNA, right? So normally nowadays scientists is more will take a research on biological molecule of protein and protein and also nucleic acid. Others actually not scientists not discover. The others is not very complicated like carbohydrate and also lipid. Lipid actually the scientist is already discover almost discover finish right. Already does not have the interesting things to discover already compared to amino acid and compared to protein and also nucleic acid. So this one are the technique that actually is just the basic technique lah, not a complicated technique. Basic technique you need to learn based on the MPS syllabus. So normally in uh, MPN syllabus just give you two techniques only you have to learn. But first technique is paper chromatography and another technique is gel electrophoresis. Just two only. Others you don't need to learn. Others you don't need to learn. Only paper chromatography and also gel electrophoresis. So paper chromatography, I think all of you already know that. I think your school already do the experiment about this. So if for those who are doing already did the experiment for paper chromatography to separate chlorophyll pigment, right? Using the Spanish plant, yeah, I'm not mistaken, Spanish plant, right? So if you already do, you should know lah the principle of paper chromatography. But for those who not yet do, you should know also before you enter the lab lah. Okay, so for paper chromatography, so actually you have to learn what is the meaning of chromatography first. So chromatography actually it is the two different words lah, chroma and graphy, color and bar. So normally it is the separation technique. Chromatography meaning that it is the separate. We need to separate the sample, separate the distribution of analyte between a mobile phase and stationary phase. So it is the separation technique. We need to separate the sample, separate the pigment, like chlorophyll pigment. So if you already did the experiment, you will find that in chlorophyll not only have chlorophyll, it have chlorophyll A, B, right? Chlorophyll A and B, xenotrophil, carotenoid, right? You are not mistaken. Carotenoid, chlorophyll A and B, and also xenophil. So so actually these four are the different components that founded in the chlorophyll one. So this one is just that we need to separate the component, the pigment. So if you see that chlorophyll, if you not separate, you will imagine that chlorophyll only have one pigment only, meaning only one component. Chlorophyll, chlorophyll. But actually, if you do the analytical method, do the chromatography, you will learn that, oh, actually, within the chlorophyll, we also have chlorophyll A, B, xanthophyll, and then carotenoid, like that. Uh, so, chlorophyll pigment is one of the application within chromatography. So, actually, in the real world, actually, we use many application not only chlorophyll only, but in syllabus you learn chlorophyll, right? But actually, you should know the theory first, before you enter the uh, application. So why we use the chromatography to separate the compound? So normally we separate the compound is based on the polarity of the component, solubility, ionic strength, or size. So polarity based on the strength of polar bond. So normally some molecule is high polar, some molecule is low polar, some molecule even no polar. So based on that polarity, we can separate them. Okay, or even solubility. Some component is soluble in water. Some component is soluble in solvent like that, right? So based on that solubility, we can also separate them. Ionic strength, some is ionic bond, some is covalent bond, some is hydrogen bond, some even just the big van der Waals force. We also can base on that, and the size. So some molecule is big size, some molecule is small size. So based on these four main characteristic, we can separate the component. So normally in your syllabus, your chlorophyll one normally is based on the polarity. Solubility, ionic strength, and size is not suitable using paper chromatography. So chromatography is the general name. The type of the chromatography you learn is paper chromatography. Actually, in you go to advanced, so normally for those who want to pursue study in biology and or chemistry, especially analytical chemistry or biology. So you will learn about the advance in chromatography. So normally I just give example: chromatography, advanced one, size exclusion, hydrophobic interaction, and then gas chromatography, and then high liquid performance chromatography, and banyak lagi. So you don't need to know that. You only need to know you for your syllabus. You only need to know the paper chromatography only. Okay, so in chromatography, you will hear about the two phase, stationary phase and mobile phase. These are the two main phase, the key component in chromatography that plays a role in separation. So what is stationary phase? So stationary phase is the either solid or liquid molecule that fix in place within a column or on a solid support, and it is the phase that play the component role in separation. So meaning that this is the place, the location that the chlorophyll pigment will be separated. So in paper chromatography, which is stationary phase? So that paper is the stationary phase. That paper, ini paper kertas ni, ini paper chromatography is your stationary phase. So I already said it is either solid or liquid material. So solid because ini paper, so it's solid lah. For liquid, like I already say gas chromatography or even high liquid performance chromatography, it is liquid material lah. But for your now, it is solid, and then solid material. So this is the place that paper chromatography is the location that act as stationary phase for the separation of the chlorophyll pigment. And mobile phase is the fluid, normally is the liquid or a gas, so fluid lah, that flow through over the stationary phase that carry the sample component to facilitate the separation. So actually, if you know that, if you directly know, if no mobile phase, so in your case, like chlorophyll pigment in your experiment you use, I think it's acetone, right? No, if not, you got acetone, right? Organic solvent, whatever, I already forget acetone. If I'm not mistaken, it's acetone or others organic solvent. Normally, it's organic solvent. If you imagine that you have to immerse your paint after you you place the dot out the paper chromatography, right? You have to draw a line, right? So like that. Better. You got draw the line at here, right? So this line is act as origin, right? And then if you did the experiment, you drop the chlorophyll drop solution, chlorophyll pigment solution at there, right? At here, right? And then after that, you need to immerse this paper chromatography into the solvent, right? So why do we need to immerse in the solvent? Kenapa nak immerse dekat solvent? Kenapa tak boleh? Oh, dot je, bagi dia separate lah. Tak boleh ke? Boleh. Kenapa tak boleh? Boleh. Maybe it requires few months lah. Why? Because it only have dot only. Dia tak ada bergerak. Dia tak boleh bergerak. Chlorophyll ni dia tak boleh bergerak. It can move or not. Kalau dia tak ada air, walaupun tak makan kipas solvent, tak ada air, is it the chlorophyll this chlorophyll pigment can be flow freely? Cannot. The chlorophyll pigment cannot flow freely. So if you want to facilitate the flow of the chlorophyll pigment, so we need the solvent, this organic solvent. So this organic solvent will act as the mobile phase. So it will flow through over the stationary phase. So dia akan bergerak kan. Ni semua solvent ni apa? If you after 30 minutes, right? After 30 minutes, you will see that you can say the liquid is when you immerse, the liquid start to flow upward until the end, right? Until the And so these other solvent is act as the mobile phase that carry the sample. Your sample is chlorophyll pigment chlorophyll pigment, and then try to flow upward through the stationary phase. So your stationary phase is paper chromatography. Okay, so this is the concept of stationary phase and mobile phase. So stationary phase, maybe it can be different. It may be normally in real case, it may be non-polar or polar. But your paper chromatography is already fixed one. So actually it's based on the polarity of the compound. Okay, okay, okay. So after that, you will get the result like this, right? You will get the result like this. The right hand side one. And then you have to require to calculate the RF value, right? So RF value actually is the value to determine the distance, and also the separation. Your separation technique is good or not, and also between to make a comparison between the pigment chlorophyll pigment. So in this case, if you see here, I think all of you already know how to calculate. So RF equal to distance of component divided by distance of solvent file. So distance of component mean that each component they have their distance. So firstly, you have to calculate the distance from original part until the end one, until the solvent flow the end part. So in this case, you see this diagram, the distance altogether is 10 cm. So the distance of solvent part is 10 cm. It's already fixed one. Just the distance of component is different. So maybe based on the cross question now, either you want red, purple, or blue. So if red meaning is 2 over 10. So 2 over 10 equal to 0.2 lah. And then this one's purple, 6 over 10, 0.6. Blue one is 8. So 8 over 10, 0.8. So this one is very simple. So I hope also in exam, if come out this question, I hope that everyone can answer it because it very simple, very easy. I can say it is bonus mark to you lah, because you just only memorize this formula. I think this formula is not very hard, right? Just you need to know that the upper part is sample, the lower part is the solvent part, and lower one is fixed one, and upper based on the component lah. So normally the high high RF value meaning that they have the strong interaction between the component and the paper chromatography. So in this case, meaning that this blue one is very high polar molecule because it have the high interaction. Because this paper chromatography normally is made up of polar molecule one. So normally this one is have the high polar molecule because it can interact very not interact very straightforward. Yeah, betul. Interact with the straightforward. Yeah. And also from here we also can determine the size of the chlorophyll pigment. So if you determine the size, normally this one is the bigger size. This one is the very small size. If you want to compare the size, blue is the very smaller size because it can flow very fast. But for the red pigment ni, dia flow sangat kecil kerana dia sangat besar. Dia sangat besar, meaning dia akan flow lebih slow lah. Compare lah macam lori dan motor lah. Kalau ini lori kan dia jalan sangat lambat kan. Motor dia boleh pusing-pusing, potong sini, potong sana, dan directly pergi sana. Kan, ini kereta lah. Like that lah. Okay. So I think this calculation I don't need to solve by because it is very easy. Okay. Now Next is about the next technique is about the gel electrophoresis system. Okay, so gel electrophoresis, unfortunately, you are not have the opportunity to carry out this experiment. This experiment you will carry out after you enter university if you got study the biology. If you continue biology, you have the opportunity to uh, carry out this experiment, gel electrophoresis. Okay, so gel electrophoresis have the three main function. The main function is to separate DNA, RNA, and protein molecule based on their size and electrical charge. Okay, so three function is DNA, RNA, and protein. It can separate these three molecule one. Okay, okay. So what are the general steps in electrophoresis? Okay, so actually it's very difficult want you to imagine. Right, because I just simple. Actually you don't need to know lah. This is just the knowledge to you because most of the school teacher also not very focused on this topic, but this topic is comes out many times in essay question. It want you to explain the principle of gel electrophoresis. So actually you need to know lah. So you should know lah actually. So what you want to prepare the gel electrophoresis is we have to prepare the gel and also prepare the this glass this prepare the sample and also prepare the gel first. And then we have to set up the electrophoresis chamber. So this is what we call as electrophoresis chamber. It look like that. Dia macam ni punya. You can imagine like that, or you can Google it image like that. And then after you prepare the gel, you can pour the gel into it. Into it. So actually, this one is horizontal one. Horizontal one. No, there are two type. Either is horizontal or vertical. Vertical dia macam ni lah. Vertical dia macam ni. Horizontal dia tu gambar lah. Macam ni lah. Also, this one is horizontal one. So horizontal, after you set up the electrophoresis chamber, you set up the electrophoresis chamber, including you pour the gel into it, okay, and then you letak itu what you call sample loading, okay, and then you have to load the sample into the vat. So why we need to letak ni comb, this one is comb. We put the comb because we need to load the sample. So dia akan ada satu lubang lah. Macam ada satu lubang macam tu. Lepas tu awak akan guna micropipette. I think this one you not yet see, never see this. So this one is one of the pipette. You don't need to know. You just need to know that this is one of the device. Okay, pipette. Okay, your pipette, your sample into the valve. What we call valve, not hole not hole, bukan lubang lubang hole kan. But we not call hole, we call valve. So dia akan ada satu lubang macam tu lah. Okay, and then you apply the current, apply the electrical current, and then it will start running lah. Power supply will start running, and then it will start running, and then it will produce the image like this. So maybe this one is not clear like this. This one is the experiment that I do during my university. Okay, so these are the three gel image that you will have to look like this. Dia macam ni lah. This is the final image lah. You will see like this. Okay, so this one is actually unclear. So normally, not only me, actually, if you first time do this kind of experiment, normally it's fail. So normally this experiment is fail. You can see that the gel, the band at there is not very clear. So actually, in this one is the successful experiment. You should get the result like that. So normally in examination, I don't know it got come out or not. So this one is the band lah. So normally the principle is DNA molecule gel electrophoresis against gel electrophoresis. Normally our in separation DNA is using against gel electrophoresis. Not only gel electrophoresis is the general name only. There are many types of gel electrophoresis nowadays. So for DNA separation, the most popular one is the agarose gel electrophoresis. So agarose gel electrophoresis. So so the principle is actually this one actually you can include in your examination one and you answer one how to prepare it after you apply the current, the separation is stuck now. After you apply the current, the separation is starting now. So normally the DNA molecule will move toward the cathode. This one will be anode place. This one will be cathode. Why? Because DNA is negatively charged, right? I already mentioned that DNA is negatively charged. You have to you uh this one's cathode because DNA will flow toward the cathode, opposite direction, right? I think this one is general knowledge for you. Like you learn physics in SPM, you should know. Cathode will flow to the anode will flow to the cathode, right? The electric current. Okay, so for after you flow, you will get the image like this. So what does this image appear give the result to you? Meaning for the component, DNA component, for those which are very larger size, it will move very slow. Dia akan bergerak sangat lambat. So if you see this result, meaning that these two molecules, this one is the DNA molecule is very smaller in size. Dia sangat kecil je. Yang ni sangat besar, very big lah, very big molecule. This is very big molecule. Big molecule. This one is smaller. So if you see here, so normally the yang paling atas tu the upper part is the bigger size one. And then the lowest part is the smallest size one. Dia sangat kecil. Why? Because in smaller size, it can move very fast. Dia macam principle chromatography lah kan, saya nak explain. Tapi never mind, you don't know. So smaller size, you can imagine dia macam motor lah. Motor boleh potong sini, potong sana kan. Dia boleh bergerak sangat lajunya. Tapi lorry, larger size lorry, dia sangat berat. So dia memang tak boleh bergerak sangat laju. So dia only can move very slow. So this is the result that you obtain. You should write in your answer. So for answer essay question. So what about? So you can make a comparison lah. So for larger size, like what? For larger size, it will move more. For larger size, it will move very slow. For smaller size, it will move very fast. Further from the valve. Okay, and then there are also the factor. So one of the factor I already mentioned is sample size. So smaller sample size will be migrate faster than larger than sample. So smaller size sample size will be move very further lah. And you have to use the migrate terms compared to use the move flow like that. I am not recommending you to use the move flow or like that. Or even not walking, I don't know. Some of you maybe walk or swim. Some of you swim. I don't know. I cannot. I am recommend you use the migrate lah, more scientific terms. And another factor is about the gel concentration. So actually, gel concentration also can affect your the flow of the sample. So if the gel concentration is very high, it will reduce the migration speed and enables preparation of the smaller DNA molecule. Okay, so normally if you increase the gel electrophoration, gel, increase the agar concentration lah. Okay, so it will reduce the speed. Dia akan kurangkan speed tu. Why? Because agar gel dia akan ada banyak lubang. If you alert agarose, what is agarose? Agarose actually is one type of the gel, and then dia akan ada banyak lubang-lubang dia. So if you increase concentration, maksudnya lubang dia akan macam jadi macam banyak. So if you walking, if you driving lah, kereta awak memandu, kalau jalan tu sangat licin punya, macam highway punya, wah awak boleh lag kan, laju sangat kan. Tapi kalau jalan tu macam kampung punya, macam lantai punya, atau ada soy, atau ada lubang sini lubang sana punya, tak sesuai punya, tak licin punya, kalau awak tak boleh mandu laju kan. So same as this, kalau if you increase the gel concentration, meaning that there are more more barrier to it, so the DNA sample will move more slower. Even though the same size, so if you compare this, if the same size, so normally the DNA same size like this, so if you increase the gel concentration, the location will be at here. For this one, it will be at here. More slower, dia akan lebih lambat sikit lah. And another one is voltage supply. Of course, if you give high voltage, high electric current, it will. Wait. Let's see the slide. Just now my internet is wrong. Okay. Just now I end at where? Which part I already lost connection? Factor affect electrophoresis. I already talk lah. Okay, never mind. Okay, I just repeat again lah. About the factor affect the electrophoresis, actually got three factors. So either sample size, gel electro concentration, or voltage. So sample size, I don't know where I lost connection. So sample size is the small sample size will migrate faster than larger sample, and if we increase the gel concentration, it will reduce the migration speed. So if you compare to although the same color, same bands at here, same size, so if you increase the gel concentration, normally the bands will be occur at here, something at here. It will move more slower, reduce the speed. Okay, and then voltage, of course, if you apply high voltage, so of course your the speed can be more faster. But normally we cannot apply too high voltage because if too high voltage, it can denature the DNA and also protein structure. So normally voltage is not the, although it is one of the effect, but we are not using that. We not using this as the main uh, procedure to increase the speed of the running. Okay, so actually about this, so type of electrophoresis. So the most famous one is agarose gel electrophoresis. So this one normally is using to separate DNA. DNA. Protein also can, but not suitable. So not suitable. So another one for protein one, normally we use the SDS page electrophoresis or paper electrophoresis. So either SDS page or paper electrophoresis. This one is more common for protein lah, but it is not suitable for DNA one. Okay. Okay, so for protein electrophoresis, so this one is more focused on paper electrophoresis. So when you using paper electrophoresis, it is more focused on pH value. It is more focused on pH value. So actually, uh, pH gradient is established in the gel before loading the sample. So actually, this one is the gel, gel. Actually, it has a different pH value. For the left hand side, the pH value is very low. For right hand side, the pH value is very high, meaning the hydrogen ion, the composition of hydrogen ion for here is high concentration, for here is low concentration. So when the protein is loaded into the middle part, and then they run the electrophoresis, they will found the band like this. So when the protein will migrate to the isoelectric pH, which they have no net charge, meaning that the protein will move toward the amino acid, will move toward uh, the isoelectric pH, meaning there is no net charge at all. So actually, if you know that amino acid, you already learned there are 20 types of amino acid, right? There are positively charged and also negatively charged, and also got some is neutral. So for neutral one, normally it will locate at the center, that akan bergerak. But for the charge amino acid, dia akan bergerak sama ada ke kanan atau sama ada ke kiri. Ke kiri atau kanan. Based on the charge they have lah. So normally if negatively charged, it will go here. Positively charged, it will go here. Okay. And then this one is about SDS page electrophoresis. The principle normally is same as just the agarose gel, but it is normally same as the agarose gel. It also separation based on the size. So it's just that the concentration is not it's just that based on the agarose gel only. So not agarose gel, SDS page gel. So SDS is the chemical component lah. Right? We call as SDS page. SDS page is the component. So normally SDS page is the polyacyl amide gel. The full name polyacyl amide gel. SDS is another name. So it's just a short form only. When we short form only. So if you compared to agarose gel, they look similar. The sample change from DNA to protein only. Ini protein kan. So protein actually got many type of protein. Either is larger size or smaller size. So same as DNA, smaller size will migrate more faster compared to bigger size. So bigger size akan trap lah. It will trap at there, and then smaller size dia small, so dia boleh bergerak potong sini potong sana, migrate more further. So that's all for the electrophoresis. I think okay, that's all. So I hope no question about electrophoresis. So So hope that you guys can understand what is electrophoresis. Okay, so I think that's all. So any question about electrophoresis? Okay, no question. Any question? You can type in the chat box. If no question, meaning that all of you are very expert. Yeah, right. Every expert say that you are not. You are. If no question, so thank you for you guys attending the class today. So if no question, thanks for your attending the class today. So I already finished the chapter 1. So tomorrow I will continue chapter 2. So tomorrow chapter actually is also considered easiest chapter, but normally most of the students are also not focused at all because it is very easy until no one to focus, no one want to focus. But when it comes out in examination, nobody can answer it. So actually, you should not behave like this. If you want to score, you should not behave like this. So for those who are interested tomorrow class, you can attend it. So for today's class is thank end. So thank you for attending. So okay, so thank you.